Hello, everyone. My name is Dr. Richard Horowitz. I am the co-host of the Healing from Lyme Summit and Doctor's Talks, and it is my great pleasure today to introduce you an old friend of mine, Dr. Garth Nicholson. Garth and I met many years ago at one of the Lyme conferences in Pennsylvania. We'll talk to you a little bit about that, of how we met and how we saw overlaps with mycoplasma and multiple infections in our patients. And it's a real honor to have Garth here today. Garth is a biochemist who made a landmark scientific model for the cell membrane. And we're going to discuss today the importance of membranes in biological function, especially biofilms and what happens to people when these membranes are damaged. And he is actually one of the first people who talked about the fluid mosaic model in medicine. And he is the founder of the Institute for Molecular Medicine in California. He serves as the president and chief scientific officer and emeritus professor. And Garth has received honorary colonel in the U.S. Special Forces. He's an honorary U.S. Naval SEAL for his contributions to our military. So, Garth, it's really a great pleasure to have you here today, my old friend. So thank you for taking the time out to do this.
Well, that's fine. Thanks very much.
Yeah. Garth, can you tell us a little bit, how did you get into Lyme? How did you get into mycoplasma? How did you get into this mosaic cell model? How did all of this happen in your life?
Well, it kind of went from one story to another story, one problem to another problem that sort of led me down this pathway. And originally, I started out my career working on very basic problems like membrane structure and so on. And we published the fluid mosaic membrane model, which is the textbook model now for membranes. But I wanted to do more. I wanted to integrate that into a medical picture. How is that important in medicine? And so I got further and further along in trying to look at not only membranes, but also other processes as well. And along the way, I did get involved in working with veterans and trying to help them and their family members, particularly after the Gulf War. So I worked very closely with Marine families here in California, and also when I was still in Texas before I took early retirement, I worked with veterans down there. And they all had some very common problems, and we'll get back to that in a moment. But some of this centered around infections and whether they had the infections before they left for deployment, or they got them during deployment, or got them afterwards. Some of the common problems, particularly as their illnesses progressed, were that they tended to collect more infections. And then we've seen this over and over again with all kinds of chronic illnesses, that people tend to collect infections over time. And so lately I've been working on mechanisms for that with a colleague of mine out here in California, Professor Safra at UC Riverside in Patton Hospital. And we've been working on barrier function and how this really impacts the numbers and types of infections that get inside your body to do real harm. As you could have superficial infections, and a lot of people see this in their daily lives. It's a common occurrence to get them in your sinuses or your oral cavity or whatever. That's not what we're talking about. We're really talking about infections that have penetrated into the blood and have spread to various organ sites where they can cause real damage. I don't think when we think of infectious disease that these superficial infections for the survival of yourself essentially are all that important. But when they do penetrate into the blood and pass to various organ sites and so on, they can be very, very debilitating.
Right. Now, we're going to talk today in detail about mitochondrial function and chronic Lyme disease. You were the first person, actually, in the peer-reviewed literature who really talked about the necessity for replacing these lipids in the mitochondrial membranes that get damaged. And also, your articles on multiple chronic infections, not only in Gulf War, but also in these people with chronic fatiguing illnesses, right? Which is, of course, what we're seeing day to day. So let's talk a little bit about these membranes, like the membrane barriers. Extracellular matrix, intracellular. Why are membranes so important? The gut, the barrier in the gut, the blood-brain barrier. Let's talk a little bit about how these get affected in these chronic illnesses.
Well, at every level in our body, we have barriers. And barriers are very important for uh, dividing enzymatic function, metabolic functions within our cells, but they're also very important in separating our organs and other tissues, and also in keeping microorganisms out of our bodies. For example, the gut barrier is extremely important, and our barriers, our mucus barriers, and so on. and our skin barriers. All of these barriers keep us, safe from invading bacteria, viruses, so on and so forth. So they're very important in that regard, and that's why it's important in Lyme patients, because any destruction of these barriers not only results in a worse outcome for Lyme patients, but they tend to collect additional infections, and that just makes their situation worse. And they can't, essentially, they can't recover if they keep having infections penetrating into their body, and they were trying to fight an existing multiple microorganism infection process like Lyme disease to begin with. So this just compounds the problem. So this is something we have to work on.
Right. So when people get like, and you and I know this because when we met for the first time, it had to be, I think, I don't know, close to 30 years ago at that Pennsylvania Lyme conference where
At least
I was
30
I was
years.
presenting on my 16-point MSIDS model, and you were presenting, and we looked at each other and it was like, oh my God, we're come up with the same conclusions on these chronic illnesses, having never met each other, right? Coming from it, scientifically just examining it. So, it sounds like we have arrived at the same conclusions over the years. So what you're saying is when people get these viruses and they get Bartonella, Babesia, Lyme, all these mycoplasma, if the barriers are not damaged, they may not actually get the same types of pathological effects. Is that pretty much the essence of what's happening?
Well, essentially, in the case of Lyme disease, of course, you get these infections injected into your system rather than recovering them through your sinuses or your gut or other ways that you get into your body. So that's one process in itself, which is very damaging. But when you're sick and your barrier functions are down because you can't replace the barriers adequately, you tend to collect even more infections. And this makes it even harder. It makes it more difficult to figure out what types of additional infections you have.
Mhm.
And it also makes it more difficult to treat all these different infections. Becomes a real problem in terms of how do you address the situation from a medical standpoint. So we've been trying to work on the barrier function as an important adjuvant of therapy of multiple infectious disease processes, because during the multiple infectious disease process, these barriers are damaged. And we can go into how they're damaged, but we've been working on, for example, the mitochondrial barrier, because that's essential in terms of generating energy. And your cellular energy is essential in driving your immune system, and every metabolic process in your system depends upon that barrier to be maintained so the mitochondria can keep producing ATP and high energy molecules. Because remember, the mitochondria provide up to 90% of your cellular energy. So if they're damaged, you're in real trouble because you can't operate, your systems can't operate in a way that can defend itself against these invading microorganisms, viruses, uh, bacteria, mold, all these different invaders cause their own sets of problems. And it
Right. So,
becomes
so what we're
incredibly
talking about is
complex.
right. So we're talking about the mitochondria being important literally in every organ of the body, right? I mean, there's no organ, whether it's the brain, the heart, everyone has to function with mitochondria. But now even in Alzheimer's, they're talking about mitochondrial dysfunction being right one of the potential drivers of dementia at this point. We know with leaky gut, the gut barrier now, it's associated now with aging. This was just published actually just a couple of days ago. We see a lot of inflammation in it. So we're really talking about these barriers. So how do you get these barriers restored? Like, let's talk a little bit about the phospholipids and how you actually do this for people when they have mitochondrial damage or they have this barrier function damage.
Well, again, first let's talk about different barriers. It's the mitochondria have their own set of barriers, and the one barrier that's incredibly important is the intermitochondrial membrane. And if that barrier breaks down, you can't generate energy. You can't produce ATP. So it maintains an electrical chemical barrier or transmembrane potential across that intermembrane barrier. And so the barrier has to be maintained to generate that transmembrane potential. And there's a number of ways that the barrier is broken down, but one of the easiest ways to break down that barrier is with oxidative stress and other means of damaging the lipids in the membrane, you create a situation where there's leakage on the intermitochondrial membrane. And when there's leakage, then you can't maintain transmembrane potential. And so energy production goes down in those cells. And of course, that leads to all kinds of problems, particularly if those cells happen to be immune cells or those cells happen to be critical cells in critical organs. You're in trouble. So your organ function tends to degrade until you can regain that barrier function, regain the mitochondrial function, produce the energy necessary to provide all the functions that our organs provide. So as people age, there's a natural deterioration that goes on. And so this barrier function actually degrades slowly over time as you age. But there are ways to try and help maintain that barrier. One of the ways that we use is to replace the lipids in the barrier, because this is one of the critical key points that determines barrier function. If the lipids are damaged, then leakage occurs across the membrane because the way the lipids fit into the membrane. And those lipids have to be replaced. So we have a natural mechanism to replace them, but it's not adequate when we have a disease process going on, because then the damage is increased so far above our ability to repair it that it causes all kinds of problems. And that's what eventually leads to cellular senescence, because it actually can turn on what's called apoptosis, or programmed cell death, and cells start to die. And before they die, of course, they're sick and they can't function properly. And this happens to every major organ in your body. There's a potential for this to happen.
Mhm.
So we know that this is an important process, and this is what ultimately will lead to a fatal outcome of a disease process. So I got really interested in this whole thing during the COVID epidemic, because we were finding that if you just had the COVID virus infection alone, yes, you could survive it. But if you had multiple infections, that lowered your chance of survival. And the more infections that you had, the worse off you were, to the point where if you had already multiple infections, like a Lyme patient, then, or a mycoplasma patient, then you were at very high risk of actually having a lethal outcome for COVID infection. So again, it added to the point, because we had seen this earlier, where if people had multiple infections, they were much more likely to have a fatal course of their disease process. And it can go in different directions. And you could have different primary problems for the actual lethal event. But the underlying problem really goes all the way back to these barrier functions: the loss of energy production, the loss of function for our immune system, the loss of function for all our critical organs, all start to break down when our barriers are broken
So,
down.
so I learned this from you years ago. So, I mean, some of the things we've used for the mitochondrial dysfunction, and of course, in long COVID now, they're showing the same mitochondrial dysfunction, of course, that we're getting in chronic Lyme and in other
Right.
diseases.
It's
Um,
literally in every infectious process it's
right,
going on to various degrees. And this is
right.
what confused, I think, a lot of people with this. Why, in some cases, you may lose a little mitochondrial function, but you can maintain. You might be sick, but you can survive. But other cases, it gets to an extreme point, then you actually succumb of
Right.
the disease process.
Now, when you use these NT factors, these glycosylated lipids, and usually when we do it, we're doing it for at least a month after dapsone, after people go through whatever oxidative stress of killing the bugs. We do it for at least a month. We're using CoQ10, acetyl L-carnitine to, shuttle the fatty acids, the energy into the mitochondria. We're also using things like MitoNR, nicotinamide riboside. We're using that right now. Other things, glutathione, to make sure they have enough oxidative capacity. NADH, right? Inata, we're using in some of these people. So do you have certain products specifically you like for this, or and how do you combine them? Like, what do you do in someone that has this kind of mitochondrial dysfunction? Because in the paper you published, I think it was, had to be at least 15 years ago, was about a third of the chronic Lyme patients, their chronic fatigue did get better with mitochondrial regeneration. So what were you using in that paper? What do you like as far as the products
Well,
that
we were primarily using really just the NT-factor lipids, which
okay.
is a formulation, which is the glycerophospholipids that form the bulk of the mitochondrial intermembrane matrix. And they're also precursors for very important molecules like cardiolipin in the intermitochondrial membrane, which is absolutely essential for function. These get damaged. Normally they're replaced because they're not damaged at a rate which would really put you at risk for having a very difficult disease process. But as you get more infections or as the infections become more severe, then you can't always replace them. So we need to help in that process. And that's why we use these lipids. And we've been going to higher and higher doses. And turns out for different types of things, like at the end of our program, I might tell you about my recent published study where we've looked at people with chemical contamination. And it turns out the higher doses of the anti-factor lipids actually encapsulate the damaging chemicals and slowly remove them from your body. And so for a lot of people that have combinations of chemical damage and infections, and this is actually very common for veterans, that you need this very high dose of the anti-factor lipids to help remove these contaminants in your body. Otherwise, you just can't recover. And this
Right.
is
So
one thing that
you're
we
talking
found.
about, for example, we're finding mold toxins now in a large number of our
Yeah, and mold
patients.
toxins fall into this category because they're hydrophobic chemical entities. And these hydrophobic chemical entities get buried in your cell and interfere with a lot of processes that go on inside your cells. And if they get in particular cells like nerve cells, they can cause a lot of havoc. And so we know that they interfere with mitochondrial function and with other membrane functions of the cell. So all these membranes of the cells are all form different barriers, and that's why the general topic is on barrier function. Once that barrier function is broken down, and again at different levels, this really leads to a disease process that's very hard to overcome unless you repair those barriers. And so that's why we say every chronically ill patient needs these anti-factor lipids, for example, to help repair all of these barriers that
Right.
are a
Now, an
problem.
interesting point, and I don't think most people know this. Biofilms can be a response to reestablish the barriers because the barriers are broken down and the biofilms are forming actually to reestablish these barriers. Is that one of the things it's doing, apart from just creating like a mechanism to give food to the bacteria, for the
Well,
channel,
it's our
quorum sensing, and
bodies
everything else that
don't
goes.
really, have a insight into some of those processes that are going on. They just naturally react, and maintaining barrier function is a natural reaction for cells. And so there are various ways to maintain those barriers. And I think that the formation of biofilm is really our attempt to reestablish or maintain barriers to not only keep those invaders out, but to also interfere with some of the things that they do to our cells. So I think it's part of a natural process that occurs.
Right.
And we also see this in terms of natural filtration in the gut. We form these barriers uh, with, you can essentially form a biofilm in the gut to kind of filter molecules out and to try and keep invaders like microorganisms and viruses out of our system. So that's really almost the first line of defense in the gut, is to form this biofilm matrix to filter out some of these invaders. And then there's another level of barrier when they get to the membrane of an epithelial cell in the gut, for example. They have to penetrate that, and there are other functions and so on that I won't go into. So these barriers occur at different levels and they have different functions, but they're all essentially important in trying to maintain our health, a healthy body and healthy function. And when the function of our various organ systems go down, you can really generally trace that back to loss of barrier function in one way or another.
Right. No, this is fascinating. Garth, if people have questions, by the way, about some of the things we're discussing today, and they want to, get questions on mycoplasma answered or the gut barrier function, where can they reach you? What is a good email to reach you?
Well, they could reach me at the Institute for Molecular Medicine. My personal email is gnicholson@immed.org. And they can reach me either through the Institute for Molecular Medicine general mail or through my personal email, and I'll be glad. I answer every email. Sometimes I'm a little tardy at doing that, but I try and answer everyone that sends me emails.
Right. Okay, that's great. Now you have new papers that are coming out at this point about some of this. You want to talk a little bit about some of the recent research you've been working on?
Well, I do have some recent research. You were mentioning the fact that it's interesting that certain diseases, particularly that are related to brain function, also these involve barriers, as it turns out. And it's very interesting. I've gotten involved in some work on schizophrenia and other psychiatric problems, which absolutely involve dissolution or loss of barrier function. And so we're going to use the lipid therapy to see what we can do with helping those patients. And we know in the case of nerve cells that the barrier function is very important and it really modifies the nerve cell activity. And we've been able to show this. So for example, that we can either attenuate depolarization of nerve cells using the anti-factor lipids, or we can stimulate it depending upon what formulations that we use. So some of these things are very intriguing, and we hope to delve into some of these psychiatric problems with using barrier repair of barrier function. They also have mitochondrial function problems, as you just mentioned already. And of course, at the same time, this helps repair the mitochondria, so the mitochondria can function, and that, of course, is absolutely essential for nerve function. So there are a number of reasons why these things are very important. And I'm just writing a review now on aging and how you lose mitochondrial function as you age naturally. So in your 90s, for example, most people have lost 40 or 50% of their mitochondrial function. They become feeble essentially because of that. So what we've done is we've taken older people and repaired their barrier function with using the anti-factor lipids, and they go on to live very active lives. And it helps them in a number of different ways. It helps in terms of their coronary function. It helps in their musculature so that they're less decline in their musculature. It helps in their nervous system and so on. So, and all of that can be traced back to loss of barrier function in various ways.
Right. So, I mean, I don't think people realize that it really is an essential function with aging. By the way, I take lots of mitochondrial supplements every day based on all the literature I read on this. But also, of course, there are natural ways of stimulating mitochondrial function, like exercise certainly is one way to do it. Resveratrol, alpha lipoic acid, right? There are other certain things also that will help to create new mitochondria, right? So are there anything else you like any other tricks you like as far as creating new mitochondria in the body, apart from, the ones we just mentioned, or it's kind of limited as you're getting older?
Well, we can create new mitochondria, but it turns out it's somewhat limited. It's not that you can completely replace all your mitochondria by taking a supplement. That's not exactly what happens. You can increase the number of mitochondria in your cells. And of course, that's important. But there are things you can do to actually increase the function of your mitochondria, or in some cases even increase the size of your mitochondria, which means more membrane, which means more function. So there are different ways to do this. And various supplements have been used that have slightly different properties and so on. And I'm for all of these approaches, by the way. As you know, we've done combinations, and we often add with our lipid products some of the very same supplements that you've mentioned. And some of our studies go back, as you mentioned, 20 years ago, we were doing this and finding that this was actually helpful. But the most helpful thing that we found are actually the lipids themselves, because they're so important in maintaining this transmembrane chemical electrical potential across the inner membrane. And if that goes down, the other supplements, no matter how you shuttle molecules in or how you do this or that, it's still not going to help with your overall ability to produce high energy molecules.
And,
So
and
the
when you're
number
talking about
one,
these, right, and when you're talking about these phospholipids, you're talking about because when for example, I detox mold, I use phosphatidylcholine and, 2.5 to 3 grams twice a day with glutathione and binders. You're talking combinations like phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine. You're talking combinations of these for the cell
right?
membranes to keep them fluid?
Yes, and in fact, our formulations are balanced for the composition of membranes, which is important. Although you'll do a certain amount of modification inside your cells, inside your compartments, like your mitochondrial compartment, for example. There'll be some enzymatic modification, but unfortunately, that can't keep up with the damage to those molecules that occurs during a severe illness, for example, severe series of infections or whatever. So it could be severe trauma, for example. And that's another thing that we found. And I've been working with veterans on gunshot wounds, for example, and showing that there's really a tremendous increase in the rate of healing if you start replacing these lipids, because the mitochondrial function is absolutely essential for the healing process and the regeneration process within tissues. So it all really goes back to things like mitochondrial function. We have to maintain that mitochondrial function to heal.
Right.
That's essential. And it's
And
healing
and the
from,
product, by the way, the product you developed years ago, just tell people a little bit about this product in case they actually do want to get it.
well, the anti-factor lipids is one of these balanced products. And the reason that that's important compared to just say lecithin or some other product is that the lipids are protected. This is absolutely essential because if you put damaged lipids in, that's not going to help you at all. You need to have protected lipids. And so we have a patented process where we use a natural product called inulin, which is derived from plants. It actually protects plants' membranes. And you find this in plants that survive freezing, for example, up in the northern latitudes. Plants use inulin to protect themselves during the winter from freezing damage to their membranes and so on. So what happens, these inulin molecules, which are fructooligosaccharides, fit in between the head groups of the phospholipids in the membranes and protect them. But also when you have the lipids isolated from membranes, they form globules and you can liposomes and various things, and the inulin will still protect those lipid molecules. And that's a patented process that we have to protect the lipids with inulins and other things as well, some antioxidants and so on and so forth.
Right.
But
Inulin
this,
is the one that comes from Jerusalem artichokes. Is that one of the sources where this is coming from? It's
well, that's one source. There are many sources of inulins. There are different types of inulins, of course, but uh, the plant world has developed all these really neat molecules to help protect themselves. And just artichoke is a convenient source, and it's very easy to isolate. But there are other sources as well.
right.
And essentially, all plants have these to various degrees.
Right.
So,
Years ago, and I learned this actually from years ago, I had a patient with cardiomyopathy who had a very severe congestive heart failure. And the cardiologists were giving him like digoxin and Lasix and inotropic medicine, anything they could do. And his ejection fraction was horrific. It was something like 19%. It was just terrible, normal being like 50 to 60. And we started him on a mitochondrial regeneration program, right, with these anti-factor lipids and CoQ10 and the rest. And interestingly enough, they rechecked his ultrasound and did a echocardiogram on him about 6 months later. His ejection fraction had pretty much just doubled, and they couldn't explain like how it had gotten so much better. And it's because obviously the mitochondria of the heart muscles were working a
yeah,
lot better. And I don't think the cardiologists, I mean, neurologists are now getting into mitochondria because Alzheimer's is now showing up as a mitochondrial disease, apart from the effects of phosphorylated tau and beta amyloid and the rest. But I don't think like every area, like GI, if you ask most GI doctors, what is leaky gut, what is membrane, unless you've done functional medicine, they don't really think much about it. And yet, as you were saying, it's those membranes in the gut, from our perspective, almost, I'd say at least 70% of my patients, 80, 90, have leaky gut. When we check them, the gut barrier
absolutely.
is compromised, and we have to use bovine immunoglobulins, get them the right probiotics. Because as you're saying, these bugs in the butt, whether it's viruses, bacteria, they're going to get in there and they're going to start causing a lot of havoc and inflammation, right? If the gut barrier is compromised.
Absolutely. And of course, again, this is something that we're very interested in. And there are a lot of disease processes that are really driven by this sort of degradation of the gut barrier. And certainly in the extreme cases that I'm sure you deal with, and we saw in the case of the veterans, because after they came home, they picked up all kinds of different disease problems. And they end up being the most severe cases, no matter what type of disease process that ultimately they have problems with.
Right.
And so this can go in a number of different directions, but ultimately you trace it back to its barriers. And
Yeah.
those barriers in the mitochondria, the barriers in the gut, the barriers in the brain, all of these are very important in different disease processes. And so this is the thing I've been trying to harp on, is that you've got to go back to some basics here. And your patients are never going to completely recover until these barrier functions are essentially
Right.
healed.
And one of the other things I've seen that interferes, we've talked a little bit about the cell danger response in some of these people where the mitochondria has, M0, M1, and there's a healing phase, and there's regenerative phase, and there's a damage phase. What I've learned before about this, though, is that we need to have more extracellular ATP, right, to kind of reverse and help. And autism, and we should talk about this because you've done papers on autism and these kind of diseases. The only molecule that I know of, I'm very curious to know if you know of any others, apart from using suramin, right, used for sleeping sickness with trypanosomiasis. I've seen some studies on John Hopkins using sulforaphane glucosinolate, broccoli seed extract, seem to help the autistic kids. Is there any other way you know of to increase extracellular ATP when you've got these mitochondria that are damaged, they're in the cell danger response, they don't move? Do you have any tricks? This was not something I was planning on asking, but I realize as we're
Well,
talking about it.
one of the things that we did early on with autistic children was to give them these lipids because it did repair their mitochondria. And once their mitochondria are repaired, then all these processes tend to be reversed.
Mhm.
And so it's something that we think is essential in terms of treating autism spectrum disorders. Is that without treating the mitochondrial function problems, they're just not going to get better. And now we're finding the same things with mental illnesses, by the way, that are being treated primarily with various drugs, which masks the problems rather than treating the problems.
Mhm.
So if you start repairing things like the mitochondria, now there's some new avenues that are going to open up for treatment of psychiatric illnesses, for example, and all kinds of mental problems and mental issues that people have, particularly when they age. And it's
Yeah.
all, I think, part of the aging process, again, is to break down the barriers. And then we end up with a lot of different problems, and you add on, and infections are just one part of that.
Right,
They're
but the point is, and you stressed this earlier, when we have these multiple infections, and of course my model is called MSIDS for a reason, it's multiple systemic infectious disease. We are seeing Lyme and Babesia and Bartonella and Mycoplasma and tularemia and Rickettsia and Ehrlichia. And we're seeing and viruses reactivating with COVID. It's happening
absolutely,
right
yeah.
all the time. You can't fix the problem with the mitochondria. If you've got active infections, right, damaging with oxidative stress, you could kind of give all the NT factors and things you want, right? But you've got to get to the sources of where the inflammation's coming from.
Well, and here's the little secret dirty trick that goes on here is that a lot of the oxidative stress is actually coming from the mitochondria itself. And that's because when you damage the mitochondrial membrane, it short circuits the electron transport chain. And then all of a sudden you get the production of reactive oxygen species right in the mitochondria, and it becomes the major source of oxidative stress itself. So there's a more than one reason for repairing the mitochondria, and that's another reason, is because then that oxidative stress goes and affects every other aspect of the cell in terms of its metabolism as well.
Mm-hmm.
So that's another little dirty secret that is going to be coming out, I'm sure.
Got it. So the oxidative stress and inflammation, it's not just from infections and toxins and lack of sleep and leaky gut and the microbiome and nutritional deficiencies, mold, heavy metal. It's actually the mitochondria themselves that are damaged that are
Well,
actually
that's
contributing in part. It's
essentially
not so much even downstream, it's also an upstream effect, right? It's happening in both directions.
exactly. When you think of the most intensive oxidative stress, it really is both. And if you can repair the mitochondria and sort of reverse that process, you can remove a lot of the problem. And by the way, I don't think people are ever going to get well unless we address this situation.
Mhm.
And because, again, the barrier function, the fact that you're going to have more infections, and also the fact that if you don't repair the barriers, the mitochondrial barrier, the cell membrane barrier, the gut barrier, the brain barrier, all these barriers, we have to maintain these barriers, not only to keep stuff out, but also to keep our metabolism intact and running like it should inside cells. So that's why I'm saying at the very simplest level, it really goes back to this barrier function that we have at various levels.
So Gar, quick question. You've published, I think, at least you're on the top of the list of someone who's published more scientific publications than anyone I know. It's over 600 scientific publications. I don't even know how you managed to do that because I think in my career over the last 35 years, maybe I've got 30 or 40, maybe once a year, twice a year, I'll get one out there. I mean, you are very prolific. You had published years ago, and I thought it was fascinating. You were talking about these multiple infections and these chronic fatiguing illnesses. Can you speak a little bit about exactly what you were finding in these people? The viruses, the mycoplasma. You're one of the mycoplasma experts. We really don't have anyone else that is like an expert like you. In fact, I brought you onto the HHS tick-borne working group committee right onto the co-infection to talk about it because I thought it was so important. Can you talk a little bit more about mycoplasma, how you think it's kind of infiltrating some of these infections? Because I don't think most people are really talking about it or how they're all
Well,
interacting.
like that's because most of the experts are either dead or retired. And I guess I'm one of the few left standing. And I've been retired now for 20 years, so I'm not even somebody that's, hanging around the lab doing lab work anymore. I mean, here I am in my home office running little small scale clinical trials and so on, but I really don't have the capacity or the organization to really delve into this anymore. But and unfortunately, I'm one of the last people standing. But I think the mycoplasma is just sort of an example of some of the things that can go wrong. And when Lyme hit, this gave us a whole new set of examples of things that can go wrong in terms of infections and so on, and how you get cooperation, not really good cooperation, but negative in a negative way, between different types of infectious processes, and how this can snowball in patients and really cause tremendous problems in terms of not only the patients themselves and their disease process, but the treatment processes and the ability to help them recover. And it gets to a certain point, and we really have to take a really kind of holistic view of these patients because they're all individually, they have their own separate problems and so on. But after a while, this business of multiple infections is very hard to ignore. And it crops up over and over in all kinds of different separate disease processes. For example, if you're a rheumatologist and work on arthritis, you don't think so much of infections, but yet infections are one of the core parts of the arthritic process. And as people that have very severe arthritis, 100% of them have various chronic infections, and they can be a different set of infections for each patient and so on, but it does cause them very severe problems. And those problems can't be overcome by simply addressing inflammation, for example, and not addressing the infections.
Right. And that may be, by the way, if you're a rheumatologist and they were treating, for example, rheumatoid arthritis and using a DMARD regimen like Plaquenil and minocycline, you and I both know that it's possible because they found mycoplasma in the joints, right, of some of these species, apart from chlamydia pneumonia and others. It's very possible it's not just an anti-inflammatory effect, it's the actual antibacterial effect, right, that
Oh,
is lowering the
absolutely.
inflammation because it's
Absolutely.
raising TNF alpha and IL-1, IL-6. It's raising all these inflammatory cytokines.
Yeah. And again, this process can really snowball. And when you have those infections, there are ways to treat it, as you know, but it becomes more and more difficult if you have when you collect more and more infections, because often there are different types of infections, viruses, bacteria, mold, all kinds of different types of infections that you can sort of accumulate over time. And it becomes more and more difficult when you take these very severe patients to take one avenue approach to treatment. You're going to fail every time because you have to address all these different problems that they have, and if you don't, you're doomed to fail. So we've
Now
been
regarding
trying to.
mycoplasma, we had talked about earlier when we were on the HHS tick-borne disease working group subcommittee for co-infections. We were talking about Mycoplasma fermentans, Mycoplasma penetrans, Mycoplasma pneumoniae, which is showing up in ticks. Do you want to talk a little bit about the fact that Mycoplasma are in ticks and kind of how this might be affecting people, since just about everybody, when you check Mycoplasma pneumonia titers, right, just about everyone has been exposed at some point in time.
Well, this is again, that's a very common mycoplasma. Some of the others you mentioned are less common, but Mycoplasma pneumoniae is the most common infection in atypical pneumonia, for example, in children. So we're exposed to this often at a very young age. And just like a variety of other types of viruses and bacteria, we can collect these, we can kind of hold them in check. But if later on in life they rear their ugly head, whether it's chicken pox or whether it's atypical pneumonia because of mycoplasma or whatever, once these things get uncontrolled again, the disease process can occur. And often it's quite different from what you've seen as a child, for example, or even as an adult, that as you age, I think you're more and more at risk for these. So I think people that succumb at before their time. Let's put it that way. I
Right.
think that this is a process that's going on, and most physicians don't have any clue.
And they, by the way, they've been finding some of these, even the autoimmune illnesses, right? I mean, in autism spectrum disorder and some of the autoimmune illnesses. I mean, we're finding a lot of these in there, and I don't think doctors are always recognizing what the role is, right? As far as driving some of the process here.
No, they tend to think of them as secondary to the disease process, but it turns out I think they're more integral to the entire disease process than people would like to admit.
Mhm.
And just like mitochondrial function turns out to be very integral to disease processes, much more than people would like to admit. That's what we're finding with these things. And so it's very hard to escape it, to get away from it, to ignore it. And of course, the medicine has been ignoring these basic processes essentially forever. And we've got to turn that around.
No, I mean, the way we address chronic disease medicine in this country, it does not work well. I mean, 86% of our healthcare costs and 70% of the deaths in this country are due to chronic disease. And we don't even have a model, like a multifactorial model like MSIDS, which I've been working on for, 35 years, to at least have a framework. I just put a study in the literature 2 days ago on some case studies of using dapsone combination therapy. And I was talking about the overlaps with long COVID and chronic Lyme, and all 16 factors on the 16-point MSIDS map are now been associated with long COVID. I mean, it's a very similar process that's showing up in these chronic fatiguing
Sure.
illnesses, but unless you're a functional medicine trained doctor, I think you're ignoring this. And I think the utility of the MSIDS model, it's like a checklist just to remind yourself, hey, don't forget mitochondria. Don't forget gut function, right, with leaky gut. Like these are things you have to remind yourself in a chronically ill patient.
Absolutely. And I'm all for it because this is the sort of pathway that we've been on for, as you know, for 35 years or 40 years. So I think we're thank God for people like you, Rick, because without people that can expand their minds a little bit, we would be in big trouble.
Well,
Uh,
and thank God for people like you. And my favorite statement about have an open mind doesn't mean your brain's going to fall out. It's an old statement, but it is true. I just don't understand in this day and age when you really do a good scientific review of like, what is long COVID? Why are people ill? What is chronic Lyme? It's not that these things have not been published in the scientific literature. It's just a question of knowing the literature, reading it, and then understanding there's a political component to this, some of these disease processes, right? As opposed to just the science itself, right? And
well, part of it
hope to
goes
get
back to medical
randomized trials.
education. This is something that I've really stressed over the years. Part of it goes back to medical education, the way we train practitioners, not only in this country, but in other countries as well. They tend to be isolated. They tend to follow certain dogma and certain routine. And I think with chronic illnesses, the way they're expanding, particularly the age-related ones, we can't rely on that system to
No, I mean,
produce
in
the
fact,
types of practitioners that we need.
yeah, I mean, the last studies I looked at, and I recently just spoke to Dr. Dale Bredesen, Alzheimer's expert in California, and the studies that, preclinical dementia, 46.5 million Americans, we're looking at over 50 million in years to come. I mean, you're talking an epidemic of a neurocognitive deficit in people where their brains are not going to function, and the mitochondria are definitely playing a role apart from infections, right? Where beta amyloid is actually trying, in a sense, to protect it. But they're finding these bugs now under the biofilms in these people with dementia, while the mitochondria are getting damaged. And interesting, I use methylene blue all the time with dapsone. They're using low dose methylene blue for mitochondrial function studies now, right? At this point in time. And it's just fascinating how the field just keeps expanding over time.
Well, thank God it is, because we do have to address these problems, because I think we're reaching a kind of a critical point where we're going to have so many people who are functionally incapable of being productive members of society, and they're going to be a drag on society, and their family members certainly would like to see these chronically ill patients do better, lead better lives, lead longer lives, and healthier lives. And that certainly is true mentally.
Right.
And again, this is something that our brains are critically dependent upon mitochondrial function, not only for activity, cognition, memory, everything you can think of that's important with our brain is dependent on mitochondrial function. So this is one of the critical elements that we have to keep in mind and keep addressing. And this is the one reason why we've been sort of pushing this business of membrane lipids. And I'll go back to a pilot study we're trying to organize in the California State Hospital Association to look at schizophrenia and other psychiatric problems, because they too are related to mitochondrial function. And we're going to try and see what we can do with that. We don't know yet, but
Hey, by the way, if you ever get that study done, one of the studies I'd love to have done is, I've always been thinking about this in the back of my mind, is to go into psych hospitals and check these people for Lyme disease and Bartonella, and even just do a skin examination to see if they've got the Bartonella striae, which are now showing up major league in the literature and in our patients with these people with neuropsychiatric disorders. One of the
well,
patients.
absolutely, along with mycoplasma,
Oh,
because this is something that I was just beginning to get involved in before I retired. That this was showing up, and of course, we found it in children with autistic spectrum disorders that 70% of them had these infections and so on. So, and even in areas, of course, the most severe cases of autism we found in areas where there was chemical exposures as well, and like agricultural areas, for example. So all these things tend to snowball.
right.
And so that we're
So by
not
the way,
only,
Garth, you've seen clinical results like using membrane lipid replacement in the autistic population who've
oh
been
yeah,
exposed to
oh
chemicals.
yeah,
You've seen the results.
oh yeah. And I originally I started with the Gulf War illness families because their children were becoming autistic. And so we were using the lipid therapy for the adults in the families, and we started using it for the children as well. And we saw really remarkable results. And then we started using it, for example, in agricultural communities where there's a very high incidence of autism spectrum disorders. And in California, we have some of these communities where they're kind of lower income communities because most of the family members work out in the fields, and they're exposed to a lot of chemicals. They bring the chemicals home, and the chemicals are in the water that the children drink, and they're everywhere essentially. And so
Well,
we have a
by the
very
way,
high incidence.
there was a study just published a couple of days ago. They're now finding microplastics in the placentas of all of the children being born. They used to find in the California studies I looked at, there were over like 50 pesticides that were showing up in breast milk. I mean, I don't think people
Yeah.
realize between the infections and the toxins, I don't think people realize like these are really major dangers to human health and productivity, and they're going to raise healthcare costs
Oh,
and
they
just cause.
absolutely are.
Yeah.
And of course, this is one reason why I've had this emphasis on using the higher doses of the membrane lipid replacement to slowly remove these chemicals from the bodies. And of course, of my published study about a year ago with Gulf War veterans who were chemically exposed to major chemical exposures in the Gulf War and have been sick ever since. And they're slowly recovering their health. And you can read my paper in the International Journal of Translational Medicine on
What,
that.
by,
It's a
by the way, what doses were you using in there to reverse some of these side effects?
six grams per day in that study, but we've gone even higher. And the latest use of this are in transplant studies where people that have organ transplantation, even higher doses than that are very important to maintain organ function in the transplant. And also it reduces inflammation and a lot of the other problems that are associated with organ transplantation. So there's just so many different potential uses of these lipids that I've been working with, and I've had a
And again,
ball
these
doing
are these are combinations. This is NT factor, like glycosylated phospholipids, not just phosphatidylcholine. It's a combination of the
it. It's
different
a
lipids.
combination. It's a combination, and they're protected with inulin and antioxidants and so on. So, and of course, then we add in other things as well. But that's the most important ingredient, it turns
Yeah,
out, is the inulin protected phospholipids of various types.
right. No, that's absolutely fascinating, Garth. So, we're coming on, well, we're close to an hour actually for our talk. So just any other points you want to make at this point, like that you think that the audience needs to understand? I think we've gotten really home some of the points that multiple chronic infections are playing a big role, as are toxins. They're getting into the membranes, they're damaging the mitochondria. Mitochondria are responsible for every organ function in your body. It's responsible for aging, for barrier function. I mean, I think we've covered anything else you think we haven't covered adequately?
Well, we kind of just touched a little bit on mental mental capacities, psychiatric problems, this and that. And I think this is an area that I want to see grow because we have so many problems that are creeping up on us, particularly in countries that have aging populations. This has become a real problem, and it's going to be a tremendous drag economically on the countries that have aging populations that have psychiatric problems and so on. So this is an area that I'm very, very interested in right now.
Mhm.
Want to push forward in.
No, I think it's extremely important. And of course, just from the pandemic itself, from people getting sick from long COVID, losing parents, I mean, the psychiatric effects have been terrible. And people like Bob Bransfield and Brian Fallon, they've been publishing for years on the neuropsychiatric effects of these illnesses. But certainly with the pandemic, it's gotten compounded. And you're right, with chemical exposures on top of the infections, we're dealing with a huge percentage of our population with mental health problems. And I don't think most people think about phospholipid replacement and mitochondrial function in these people. So I think you're bringing up an important point of research for the future.
Yeah, again, it all goes back to barrier function, which is what I started our conversation on, because the barriers at various levels, at the mitochondrial level, at the cell membrane level, at the nuclear level, at the functional level of organs, at the barrier, the gut barrier, the brain barrier. All these barrier functions turn out to be very integral.
Right. Garth, do you have any favorite tests you like? I mean, apart from you can go to LabCorp and get a CoQ10 level and acetyl L-carnitine. And I know they're in Europe, they have a bit more testing actually for mitochondrial to look at, what's going on. Do you have any favorite tests you like actually for testing mitochondrial function?
Well, unfortunately, that the types of tests that we've preferred to see are ones that are too difficult to really do commercially, or
Mhm.
at least that there's kind of an impediment. Commercial tests, they tend to prefer simpler tests that they can reliably perform and maintain their laboratories in terms of performance uh, for routine testing. So routine testing is a lot different from, as you know, from research testing. Now, research testing, we're not limited more or less in some of these, more, I guess, everyday problems that crop up.
Right.
But that's one of the problems. I think we need to somehow take our research tests and rethink them so that we can develop simpler tests that can be done commercially. If
Mhm.
this is a real impediment, the
Right.
research is going way, way far beyond what can be done practically in a commercial laboratory. And having been involved in running a small commercial laboratory at one point in my career after I retired, I know that this is a real problem.
Right. So, but if you check, for example, lipid peroxides, F-isoprostanes, I mean, there's certain things you could check, 8-hydroxyguanine, protein carbonyls. I mean, there's signs of oxidative stress, right, that you
Yes,
can measure,
there's
which
certain, yes,
that
there's
would be
certain
like an
things you can do.
indirect marker of abnormal mitochondrial function.
Yeah, exactly. But to really do, good study on mitochondrial function, I'm afraid you can't get that done commercially, unfortunately.
Right.
And because you have to look at various aspects of mitochondrial function, and that just can't be done in a commercial test because it becomes too expensive. And that you can't get reimbursed for expensive tests, essentially. That's the bottom line.
Right. Yeah, it's usually specialized hospitals, Mayo Clinic, they'll check mitochondrial, right, DNA and different changes. In fact, I have a young woman coming in the next week or so. She has a mitochondrial disorder, a genetic mitochondrial disorder, and has Lyme. And so far, they've not been able to get this woman better. And she's going to be coming to see me, where I'll be using obviously a lot of the mitochondrial things we're talking about today, apart from getting rid of the infection. So it should be fascinating to see how this young girl, she's only 18 years old, how
Yeah, and this is kind of what we've been talking about in general, is that you have these various aspects of disease, they tend to be compartmentalized. And that's sort of what, I was back and forth mentioning, but not directly addressing, in our conversation today, is that they all have impact on each other. And it's very difficult for a given uh, practitioner or even a team to approach because it involves so many different aspects of their metabolism, of various things like infections, uh, immune system problems, all these different things take different specialists in a way to do it. So you almost have to have a team effort to really address these difficult problems.
No, and it's why I'm actually happy I ended up becoming board certified in internal medicine, because in internal medicine, you had to know a lot of things, right? At least a little bit about a lot of
A
things, and
little
enough
bit about
that
a lot of things, right?
enough that you could kind of dive into some of these things and have some knowledge of it, right? As opposed to subspecializing in just one area. And it turned out to be, of course, very useful for this chronic fatigue population with tick-borne infections. So yeah,
Well,
so
and
Garth, we've reached our hour time. I just want to thank you so much for taking the time. Can you tell people again, if they have any questions, best email to get in touch with you?
it's G Nicholson without an H. It's GNICOLSON at immed.org, or my home is at cox.net. So
Great.
either of those two email addresses, they can get in touch with me.
Great. Thank you so much. Garth, it was so good seeing you. You and I have been, we've known each other for a long time since we met at that conference. And, you're on the MSIDS board with me, and you've been a tremendous, real boost for the scientific community with your work on barrier function and mitochondrial, the cell membrane fluid mosaic model. I mean, thank you for all your contributions to science. You've been really a pioneer, and
Yeah,
I'm appreciative for it as
we
many.
passed the 50 years of the fluid mosaic membrane model, and it's still in the textbook. So I think it's there for good.
Yep. No, you're the one who got it in there. Anybody who reads a biology textbook so learns about the fluid mosaic model, it's you. You're the one who started it. So it was a real honor to speak to you today, and I'm glad we had a chance to talk. So again, for those of you who've been tuning in, my name is Dr. Richard Horowitz. I'm co-host for the Healing from Lyme Summit. We've been speaking to Dr. Garth Nicholson about mitochondrial dysfunction and barrier function in those with chronic Lyme and chronic fatiguing illnesses. Again, thank you for joining us all today. We hope you'll be tuning in to another episode very shortly. Thank you.